So, you ever looked at a sick plant and just wondered, what is going on there? Well, it turns out it's more than just a sad looking plant. It's a mystery waiting to be solved. And the scientists who crack these cases, they're part detective, part biologist. Today, we're going to follow their checklist and do a little investigating into the world of bacterial plant diseases. Okay, so here's our crime scene. We've got a plant, and it's clearly in distress. I mean, look at the evidence. The leaves are browning, the stems are wilting. Something is definitely wrong here. But what's the cause? Is it a fungus, a virus, or could it be some kind of microscopic bacterial culprit? That's what our investigation is all about. So, how do you solve a plant crime? It's not about lucky guesses or just, wild theories. No. It's about a systematic, step-by-step investigation, one that takes you from the field to the lab and finally to your prime suspect. Every good investigation begins right at the scene. For a plant detective, this means getting your boots on the ground, out in the field, observing absolutely everything, and asking all the right questions. This is where you find your very first clues. And this right here gets to the core of it. Plant diagnosis isn't guesswork. It's a science of deduction. You have to follow a series of logical steps without jumping to conclusions. You just follow the evidence one piece at a time. The first clues are always the symptoms. The detectives are looking for those telltale signs. Here on this soybean leaf, see these dark, kind of spreading lesions? This is known as bacterial blotch. It's a classic piece of visual evidence. Other times the evidence is, well, a lot more dramatic. This poor pepper plant is suffering from soft rot. The bacterial enzymes are literally dissolving the plant tissue, turning it all into this mushy, decaying mess. It's a very distinctive clue, that's for sure. And on these potatoes, we're seeing another really common symptom, scab. These rough, almost cork-like lesions on the surface are a super clear indicator that something's wrong down below the soil. Each symptom is a different clue, and each one points towards a different group of suspects. So the detective's field checklist is simple, but it is absolutely crucial. First, you identify the victim. What kind of plant is it? What's its history? Then you document the symptoms, and I mean meticulously, looking at plants in every stage of the disease. And finally, you do your background research, checking the records for any known diseases that affect this crop in this specific area. All right, with all that field data collected, it's time to head to the lab. I mean, after all, you can't exactly interrogate a suspect you can't even see, right? The next critical step is to find those bacteria and isolate them from the plant tissue, getting them out of the plant and into a petri dish. And one of the very first tests they run in the lab is, well, it's beautifully simple. It's called bacterial streaming. If a plant is dealing with a certain type of wilt disease, you can cut the stem, place it in water, and get this stunning result, a milky, almost ghostly cloud of millions of bacteria just oozing out. And just look at how simple the setup is. This is a test you could practically do in your kitchen. You take a cut stem from a wilted tomato plant, suspend it in some clear water, and the detective just has to watch and wait for the evidence to appear. And this is what's happening on a microscopic level. We're not talking about just a few bacteria here. This is an enormous population making a break for it, escaping from the plant's vascular system. This is our first direct visual confirmation that bacteria are present in massive numbers. But, and this is a big one, it's important to remember this test isn't foolproof. Just because you don't see any streaming, that doesn't mean you can rule out bacteria as the cause. Sometimes other particles can get in the way, or maybe the specific culprit just doesn't produce a stream. A good detective knows the investigation has to go on. Okay, so once the bacteria are isolated and they're growing on a nutrient medium in a petri dish, now the real interrogation begins. To identify our suspect, we need to build a complete profile, and we do that by running a whole series of biochemical tests. The very first question is a fundamental one. What kind of cell wall does our suspect have? See, bacteria are divided into two major groups, gram-positive, which have thick cell walls, and gram-negative, with thin ones. This one simple fact is going to narrow down our list of suspects a lot. So to figure this out, scientists use what's called the KOH test. They take a drop of potassium hydroxide and mix it with the bacterium. Now, if the bacterium is gram-negative, its thin little cell wall ruptures, spilling out all of its DNA. And that makes the whole mixture, well, gooey and viscous. And here you can see the results in action. On the left, it's just a watery suspension. That tells us the thick cell walls of gram-positive bacteria are holding firm. But on the right, whoa, look at that! You can lift a stringy mucoid thread. That's the released DNA, and it's our confirmation. Our suspect is gram-negative. But the interrogation keeps going. Can the suspect grow in high salt? What about at different temperatures? In this test, the cloudy tube on the left, that's called turbidity, shows positive growth. The clear tube on the right is negative. Every single result is another piece of the puzzle. And there's a whole battery of tests that builds the suspect's profile. Can it use certain sugars for food? How does it move around? Does it have flagella? Can it cause rot in just a simple slice of potato? Every single test reveals another key characteristic, helping us build its unique identity. Okay, now we have all the data from the lab. We are ready for the final step. We've got the field report, we've got the lab results, all the evidence is right here on the table. It's time to make the final identification. This final step works just like cross-referencing a fingerprint in a criminal database. Each of these columns represents a different known pathogen. So the detective takes their test results. Let's say they got a negative on the PHB test, they saw 1 to 6 flagella for movement, and a negative on the oxidase test, and they just look for the one column that perfectly matches all the evidence. And when you find that one perfect match, you've got your culprit. This unique combination of biochemical traits, it acts just like a fingerprint. Through this methodical process, from the field all the way to this final chart, the detective can finally name the pathogen with certainty. Case closed. And that, my friends, is how you solve a plant crime. It seems even the bacteria we've been investigating wanted to help us explain the process, so thank you. But of course, identifying the culprit, that's really only half the battle. Now that we know who did it, the next big question is, how do you stop it from happening all over again? Now that's a case for another time.